Hello. Before we go into gene regulation, we need to take a little look back on transcription control. So we'll take a look at that first, but I think I better get a sword first because there are creatures out there that could potentially cause me a few problems. So we'll go take a look at our transcription again. Fly out of the house. Remember, these are Dr. K productions. And we'll head over to the transcription area. Remember, transcription is RNA being made from DNA. And that RNA is going to be translated into protein. This happens on RNA polymerase. And this shows the DNA molecule we showed once before, showing the complementary base pairs. And those complementary base pairs running all along the DNA molecule. Where the arrow is the start of transcription. And the DNA feeds into the polymerase. The polymerase itself moves along the DNA, splits it open, and when it splits it open, you have one strand, which is the coding strand, that's going to match what the RNA is going to be, except for T being transcribed into U. In the center you see the relationship between the bases. The whole system moves in the direction of the arrow. And then you have RNA made off from the non-coding strand, and that RNA is synthesized, comes out of the polymerase, goes out of the nucleus into the cytoplasm, where it's made into protein through translation. One of the things you see here, see the light-colored blue box, those are representing T's, the dark-colored blue representing uracil. So the sequence itself is the same, except for the fact that T is replaced for uracil on the coding strand. Once that synthesizes out, you have the 5' cap, and when it runs out, the whole process is going to go, again, outside the nucleus so that you can start translation. So now that we know what transcription looks like, when we talk about gene regulation, I'm not going to show the individual base pairs, and we're going to do it just a little bit differently, but keep this whole transcription thing in mind. And if I can find my house, I can sleep, and then it will be daytime where we can take a look at gene regulation in the little Minecraft world. So we'll head back to my house, go in, take a little bit of a nap. Well, first we ought to have some entertainment here. Oh, got him. Uh-oh, he's going to blow up my Dr. K. I got to take care of this. Hopefully he doesn't get me. Oh, made a little bit of a hole out in the sand, but now it's time to go night-night. So we'll head into the bedroom back in the library and have some sleep. Boom, we wake up, it's morning, and let's head out to do some gene regulation. Now, in terms of gene regulation, what ends up happening, if you look at the molecule that's laid out here, imagine that being a loop of DNA coming out of the chromosome somewhere. And what's going to happen is you've got a group of genes in an operon, which are all grouped together. On the left side, there's a repressor. On the right side, there's a terminator. And here we have 4 completely different genes represented by the colors. Again, I'm not showing base pair matching colors like we did before. This time the colors represent the genes themselves. So although it's showing it as a double strand, that could be thousands of bases long. This is a repressor bound to the operator region. And what that does is that prevents the RNA polymerase from coming in and initiating RNA synthesis. So as long as it's sitting there, it will prevent any synthesis from this operon. So this is like the lac operon that would prevent the beta-galactosidase gene or the permease gene or any of the other genes related to lactose metabolism from being synthesized. So think what I'm going to do is I'm going to bring in an inducer. An inducer will take and release the repressor from the operator region. So I'm going to leave my house. It's kind of scary out there, but I actually decided to dress up like one of my explosive friends. Oh, get out of there, spider. All right, so let me head over there, and we'll go over to the repressor, and so I'm going to deliver the inducer. The inducer is going to be whatever it takes to get rid of the repressor, and once the repressor is missing, the RNA polymerase will bind and start to make RNA from these genes. Again, these may be representing thousands of base pairs, the different colors showing different proteins that are made. So here, I'm going to throw some inducer around. So this might be some lactose coming into the cell and getting over next to the repressor. There's plenty of inducer there. And what that's going to do is it's going to cause the repressor itself to fall away from the DNA strand. And here goes the repressor. Okay, so now the repressor's gone. RNA polymerase has the opportunity to come in and start to synthesize RNA because there's nothing blocking that synthesis now. The DNA, the operator region, the promoter region are available, and here goes the synthesis of RNA. Again, we're not matching base pairs like we did before. I'm just putting the RNA down to represent the synthesis from that gene. It means it's making that gene product. It means it's going to make, if it were LAC-Z, it would be the enzyme that breaks down lactose, and then LAC-Y, which would be the permease, which allows lactose to come in at a much greater concentration. So what this does is this turns on that operon, and that series of genes is now operational. I'm only showing one RNA being made, but it would be repeated, and there would be thousands of RNAs that would be made. So now, what happens? This RNA is going to go out into the cytoplasm, be made into protein. It's going to metabolize the lactose if we're using the lactose operon, and eventually the lactose is going to disappear. And when that lactose disappears, what's going to happen is this gene, which is still running full bore, I'm only representing one RNA being made, but it would make more and more. It would make all kinds of protein, and you'd be loving eating lactose as much as possible. And then eventually lactose is going to start to run out. So as lactose is being depleted in the cell, the repressor itself is going to go back, reattach to that operator region, and once it reattaches to the operator region, it's going to shut down transcription again, and that is how a gene turns on and off in an inducible fashion. It turns on by removing the repressor from the operator, and when the repressor goes back to the operator, it shuts it back off again, just like turning off the lights in your house.